My name is David Haselbach and I'm a group leader at the Research Institute of Molecular Pathology in Vienna. And my group is concerned with how do you determine the concentration of proteins in a cell, so how the cell makes sure that the protein concentration is at the right level. And this can be done through a delicate balance between the protein synthesis that is done by the ribosome, that you see here, and that can also be done by protein degradation, which is mostly done by the proteasome, which you see here. You have the possibility to increase the protein synthesis to make more of your protein by just increasing the translation rate, but you can also do the opposite. You can also decelerate the ribosome, reduce the number of ribosomes, and get less of the protein by maintaining or changing the activity of the ribosome. On the other end, you can do both things also with the proteasome. So you can increase the concentration of your protein by lowering down the activity of the proteasome, or you can make the proteasome more active, and with this you will get lower concentration of the protein that you really would like to have. In most cases, we have a delicate balance between all of that, so we really want to maintain a certain concentration for a certain protein in a special environment, and we have looked at how this balance is kept from the side of the ribosome on the one side, and there we have studied how ribosomes are made and how ribosomes can be held dormant with our collaboration partners. And on the other side, the main focus of my lab is to understand how the proteasome is maintained and how the proteasome is regulating the protein concentration. Just a quick look at the proteasome, because most of my talk will be concerned with that. Just a quick reminder, the proteasome is made of two parts, which is the core particle, which does the job of the degradation, and the regulatory particle here on the top, which recognizes the substrates and determines whether a protein will be degraded or not. There is a number of possibilities to regulate the proteasome, and the cell can use all of them in parallel. The most easy, understandable possibility is changing the abundance of the proteasome. More proteasomes will degrade more proteins in less time. Less proteasomes will degrade less proteins in more time. An alternative to regulate, and this is less well studied, is the localization of the proteasome. The protein of interest and the proteasome have to come together to be degraded. If the proteasome doesn't see the substrate, it will not lead to the degradation of the protein of interest. So we really have to make sure that these two parts come together. A third possibility, and that's important to understand, is that there's not the proteasome. There is thousands of different possibilities of proteasomal assembly. Additional to the parts that I just told you, the regulatory particle and the core particle, there is a lot of accessory proteins and other adapters that can bind the proteasome and modulate its function. The main regulation of protein degradation is, however, the targeting of the substrates through E3 ligases, which will ubiquitinate the substrate, and also they are regulated, counter-regulated by deubiquitinating enzymes that will lead to the survival of the protein. For the main part of this talk, we will focus on the localization, and we will focus on one very special localization, which is the nuclear localization. When you look in most of our cells, of our human cells, you will notice that in most of these cells, the proteasome is actually nuclear. About 90% of all proteasomes in our cells are in the nucleus. Why are they there? Because you have transcription factors and other regulators in the nucleus that have to be degraded really rapidly. They are made really rapidly, but they also have to be degraded very rapidly. Otherwise, you cannot react to any kind of stimulus in the short timings that you will see in cells. While this is very clear, there is one problem that happens, especially in vertebrate cells, is vertebrates have open mitosis, meaning that the nuclear envelope dissolves during mitosis, releasing all the content of the nucleus, and which you see in this one cell here, that all the proteasomes are outside of the nucleus. So once mitosis has finished, all these proteasomes have to come back. To understand how the proteasome is then imported again, let's take a step back and look how actually a protein is normally imported in the nucleus. What you will see how here now is a general protein, is a nuclear protein. And normally, if this hits the nuclear pore, which is the entry gate to the nucleus, it will not be able to enter the nucleus because the interior of the nuclear pore will just repel it. But once it binds nuclear transport receptors, these nuclear transport receptors can package this protein and bring it to the nucleus. In the nucleus, this complex is disassembled and the protein stays in the nucleus and again doesn't come out anymore. What do you have to do for larger cargos? Large cargos, on the one hand, and this is a study from the Görlich lab in Göttingen, either have certain surface properties, meaning they are very hydrophobic on their surface, which basically no protein is and none of the cargos is, to enter the nucleus. So this is not true for the proteasome. With this, we cannot get into the nucleus. So it would actually need nuclear transport receptors. And then the question is, how many of these do you need? And this is a study from the Lemke lab in Mainz, where they used a 1.5 megadalton complex, not the proteasome, this was a VLP, and showed that actually these 1.5 megadalton complex needs tens of nuclear transport receptors bound on nuclear localization sequences to actually be able to go into the nucleus. So what we have to figure out now and what we have to find is, how does the proteasome actually binds tens of receptors to go into the nucleus if it would follow the standard model? What do we know already? We actually knew quite a lot from the work of the Baumeister lab in Munich, and we know that proteasomes are imported into the nucleus intact. So they're not disassembled to be small proteins, they have to go intact. Then we know the proteasome already has nuclear localization sequences, and it has 8. Getting back to the model that I just told you, 8 is not enough. So we need more. What the Baumeister lab also found was that there are components in the cytoplasm that are sufficient to transport the proteasome in, but they just didn't know which were these components. And finally, and very astonishingly, if you take proteasomes from archaeal or actually bacterial proteasomes, they will be transported into the human nucleus if you inject them into the cytoplasm. So all of that was the perfect starting data to ask the question, how is it now transported in with all this preliminary data? And this raises the question, what is necessary for the nuclear import of the proteasome? And we answered this question fairly serendipitously, because Johannes Zuber's lab here in the IMP was doing a CRISPR screen to figure out what regulates MYC. And in this CRISPR screen, they found a protein called Acarine 2, which seems to be one of the major regulators of MYC. They didn't really know what it was, so they came to us and asked, does that have anything to do with the proteasome? And we actually didn't know anything about this protein and got very intrigued. So we teamed up to figure out what this protein does. And to already tell you in a nutshell what Melanie von Hannes' lab did actually do is she looked into the phenotype of the knockout of this protein. And what you see here is we labeled the proteasome and looked at the proteasomal localization in the wild type. And you see the proteasome is released during mitosis. All the proteasomes are in the cytoplasm. Mitosis goes to the end, the nuclei reform, and proteasomes are back in. That's the wild type. Once we knock out Acarine 2 in this case, we see the proteasomes are again released into the cytoplasm, but they never come back. So these nuclei remain empty, meaning that Acarine 2 is a necessary component for the nuclear transport of proteasomes. So this was the component that was in HeLa cytoplasm, but no one has found so far. And now the question remains, how does the nuclear transport complex look like, and how does Acarine actually do this job? First, let's have a look at Acarine 2. Acarine 2 is a protein that is majorly unfolded and has just 2 parts that are folded, which are a coil-coil domain on its very end and a small NLS, which is also an alpha helical element. Everything else is disordered. It's not ideal for structural biology, and we're a structural biology lab. Nonetheless, we took the challenge and made a complex out of Acarine and the proteasome and did cryo-electron microscopy with it. And what you see here is actually the structure of the proteasome. This is a half proteasome and Acarine. Acarine in red sits on the top of the proteasome, actually silences the proteasome to not be active anymore because the entry for the peptides to go into the proteasome is just below this coil here. And Acarine is actually sitting into pockets that normally the activating subunits, so the regulatory particle, would use to activate the proteasome. So Acarine is necessary to silence the proteasome during transport. The revolution that happened in the last 1 or 2 years is that AlphaFold was there, and we could predict structures very well. And we did that as well with Acarine. And this is the prediction for a dimer of Acarine. And you see that this coil forms a dimer very nicely, and this is the part that we see. The part that we don't see is all the rest, and you see all of this is flexible parts and not really well structured parts. They will flop around in the solution, and we don't know what they do. We know that this coil part will silence the proteasome, but we don't know what the rest is doing, which is very essential for the nuclear transport. And then we turned again to AlphaFold and asked the question, if we now search for all the proteins that we know by mass spec analysis actually bind Acarine, which ones of those would actually predict to be a complex? And this is the result. So all the mass spec results we put together, and what you see here is on the one hand, how well can you predict the structure of the protein of interest and how well does this protein interact with Acarine. Unfortunately, none of the proteasomal subunits are predicted to bind Acarine at all, even though we have the structure. But interestingly, there's a couple of other proteins that very well are predicted to bind Acarine and also the proteasome, which is on the first place importin alpha. There's an autolog Acarine 1 that also can make dimers, and there's another importin called importin 9 that is essential for the transport. Which of these structures can we really look at and predict? And the main structure that we can get out of AlphaFold is the importin alpha Acarine structure. And what we see is that importin alpha is actually binding this flexible part here. Here's the coil that we have seen before, and this is on the very other terminus that nicely binds into this importin. So what we can imagine now is a complex of the proteasome, Acarine sitting on top, and the flexible part being in the solution binding one of these importins. But again, we still have to bind tens of them. So one is not enough. So we wanted to see the entire complex. For that, we did mass spectrometries. We made pull downs with Acarine again and wanted to see what is actually binding Acarine very strongly. And this is the full spectrum of everything that binds. And you see everything in blue is proteasomal components. And you see basically the entire proteasome is binding. And we see a couple of other components that are already mentioned. There's importin 9 and there's standard alpha importins. And a small GTPase that is also important for nuclear transport is called RAN. We made all of these components recombinantly and put them together with our proteasomes and see which of them co-migrate on a gradient, and actually all of them co-migrate very well. So all of these components build a large complex, which is really nice for us because the larger the better in cryo-EM, so we can make a structure of that. And this is how it looks like. So we see here the entire 20S proteasome. On top, now in orange, we see Acarine, and this is a dimer of Acarine that we see in the front. What we see here in blue is IPO9, and in the back, we see another dimer of Acarine. So what we now have is we have the core particle and 4 Acarines and 1 nuclear transport receptor. Remember, the flexible parts that we don't see in our structure also bind importins. So what we will now have in this complex, we will have 5 importins binding the whole complex, still not tens of them. So we still have to figure out how to go on with that and how do we get more of these proteins binding the proteasome. And for that, we looked into the properties of Acarine, or the physical-chemical properties. And we noticed that Acarine is clustering. So several proteins of Acarine cluster together to form larger amounts of them that might be then on top of the proteasome that actually would lead to clustering of more nuclear transport receptors on top of that. And this we have done by labeling the well-type Acarine with GFP. And what you see here, it forms droplets in basically any condition, and you can change the buffer, the pH, and it will always cluster. So that's a horrible protein to work with, but this is a property that is very inherent to Acarine. And what it makes it actually even worse is if you add IPO9 to the system, which we know binds it all. And we see here already that these clusters get very, very ugly and actually get precipitates. So they get larger and larger, such that they start precipitating and cluster more of the nuclear transport receptors. And you can also appreciate that in EM, where you see if you don't have IPO9, you can still recognize individual particles relatively well. Once you add IPO9, there's always a mess and fairly hard to analyze. And the reason for that is that one IPO9 can at least bind 2 dimers of Acarine. And if you think that through, you can make a large complex of it all. Now, this is beyond the abilities of single particle EM, because all of this in higher concentration is a mess, and we cannot see these large complexes in single particle EM. So what we wanted to do is, can we see these complexes in the cell? And it seems to be like the search for the needle in the haystack. If you just imagine that you just have to find one proteasome being in the nuclear pore at a random time point where the proteasome is transported into the nucleus. But that's not what happens. What happens is that the proteasome is actually only transported during the time frame after mitosis has ended, and this is just 10-15 minutes after that. So what we have to do is we just have to look at our cells and on our nuclear pores once mitosis has ended, and for this we look at the cells in light microscopy, as you see here. And once the cell has divided, we rapidly plunge freeze the cell and do cryo-electron tomography with that. So what is cryo-electron tomography? How do we investigate the proteasome? So what we have to do is we grow our cells on grids, on electron microscopic grids. Once we see the cells have divided, we plunge freeze them in liquid ethane. Then we have these cells on grids, and we put them in a focused ion beam machine. In the focused ion beam, we make very thin slices, lamellae, from the cell that we can then analyze in a transmission electron microscope. In a transmission electron microscope, we take a tilt series, so we look at the sample from different orientations, and from that, we can reconstruct a 3D volume, recognize all these proteasomes, and can average them and see how these complexes look like. How does that look in reality? So this is one of the tomograms that you see here, and just to guide you through, here on the right-hand side, we see the nucleus, and on the left-hand side, you see here membranous compartments, which is the ER in this case. And in all this gray noise, you might recognize small little elongated shapes here on this side, which are proteasomes, and just for you to appreciate more, I magnified them out. So you see this is how these proteasomes look in these tomograms, and we can actually understand how these complexes look like and how they assemble to these larger complexes. Now, in the next step, the mantra of the structure biologist is always structure equals function. How do we add function to that? This is very tedious in most cases, because what we would do is we would look at the structures very intensely and find the individual amino acid contacts that we think might regulate the function. We found that too tedious to just think about that and do rational design. So we started again to collaborate with the Tsuba lab and try to look at all different amino acid exchanges that you can do. Acarine is just 202 amino acids, so it's fairly feasible to do all possible amino acid exchanges. So what we do is we knock out the endogenous acarine and we replace it in cells by all possible point mutations. We then look at different properties in which we look for the MUC level, which was what we initially looked for. So low proteasomes in the nucleus, meaning high MUC levels. We looked whether these cells survive, because without proteasomes in the nucleus, these cells die. And we can actually directly look at proteasomal localization in the nucleus with a specialized fax machine. The data that we then get is whether these cells survive or one of these traits is high. And then we know for every amino acid, for every possible amino acid exchange, how important is that for the function of the proteasome. And what you see here, and this is a control that we already knew, that the very last amino acid of acarine is absolutely essential. So whenever you stop acarine before that, it will not work entirely. So that's why you see every exchange to a stop codon is lethal for the cell. Then the question that remains is, how is acarine actually released? So how do we get rid of acarine in the nucleus? And for that, we looked into different possibilities. We knew that acarine is actually turned over really rapidly. It's only expressed during mitosis, and once mitosis is over, it's gone completely. We did experiments to see actually that when we put a proteasome inhibitor, acarine really accumulates, so it's proteasomal degradation that happens. But if we add a ubiquitination inhibitor, acarine is still degraded, means acarine is degraded in an ubiquitin-independent manner, which is no surprise if you think that acarine is a fairly unfolded protein, and unfolded proteins are rapidly degraded by the proteasome. And this we can all reconstitute actually in cells and see the same result. Once we just put acarine together with the proteasome without a proteasome inhibitor, it gets degraded. In this case, we have a fusion of acarine and GFP, and then the proteasome can only degrade the acarine portion, and GFP remains, and this is what you see here left on the gel. We know that acarine is ubiquitinated by the E3 ligase UBR5, and we see that UBR5 is actually binding acarine, which we can see here in gradients. UBR5 is an essential E3 ligase that is exclusively nuclear, which is essential for a number of cancers for the cancer cell to survive. It ubiquitinates acarine, and it ubiquitinates a number of other factors, but mostly in a secondary manner that it prolongs chains to actually decrease the lifetime of proteins. And we have just determined also the structure of this E3 ligase, actually in presence of acarine, but as acarine is very flexible, we actually don't see any trace of it and don't yet know what it does. So to sum up, what do we know about the nuclear transport of proteasomes? That's our current working model, and we have our proteasome. The proteasome is binding acarine. Acarine is recruiting and clustering a lot of nuclear transporter receptors on the top. The whole assembly goes into the nucleus. In the nucleus, this complex is disassembled by the action of RAN, and actually acarine is degraded directly by the proteasome. I have just shown you one example of how the proteasome is regulated in the cell. However, this was just one of the 4 possibilities to regulate the proteasome, and in reality, all of these 4 different things can happen at the same time in the cell. So we have all of these processes in the cell, and we have huge proteasome heterogeneity to maintain the concentration of individual proteins. So what we look into now is how all of this acts together in the larger context of certain cellular models. And to give you 2 examples, we're looking into spermatogenesis, where the proteasome is actually transported into the nucleus, is going out again, changes its localization, changes its assembly state with a number of factors that I'm just showing you here, and we have found one of the factors sitting just on the top of the proteasome regulating it, and this is necessary for spermatogenesis. And this is one of the things that we look into in the future, just showing you here that all of the factors I actually named, plus this additional one, will play a role in spermatogenesis and will play a role in male fertility. Alternatively, and also important, is the role of the immunoproteasome in antigen presentation. And the immunoproteasome is exclusively sitting on the ER. So what you see here, it's not in the nucleus, while the standard proteasome is in the nucleus and in the cytoplasm. When we have immunoproteasomes, we have them in the cytoplasm on the ER. And this is something that we have to understand, because the immunoproteasome is actually not as different to the standard constitutive proteasome. And with this, I would like to thank the people in the lab with which I work, and we have a number of collaborators that make all of this possible and give us the opportunity to do cellular work in different organisms, and I thank you for your attention.